Thank you everybody for joining us today for the IWGSC webinar series. My name is Joshua Stein. I'm a project manager with the IWGSC and will be hosting today's talk. Oops. Oop. I just want to give a few words about the IWGSC. We are the International Wheat Genome Sequencing Consortium. We were formed about 20 years ago as a not-for-profit organization, and with the goal of getting an international community around building tools and resources for wheat genomics. Currently, we have about 3,500 members from 80 countries and 985 institutes or companies. And in addition, our very special members are our sponsors. We have 6 sponsors that together make this webinar series possible. So let's thank them. We have Arbor Biosciences, CIMMYT, Florimel Depre, INRAE, RAGT, and Kansas Wheat. So thank you, sponsors, for your contributions. So most of you probably know that the IWGSC's principal goal was to produce the first reference genome sequence for hexaploid wheat, and that was accomplished back in 2017, 2018. And since that time, we've started a new vision for IWGSC 2.0, and that is to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. And we have been working now for almost 2 years on this new project, which is funded both by the National Science Foundation and by the European Union. The idea is to preserve, discover, and mobilize genetic resources by sequencing the landrace genomes that encompass the worldwide genome variations of bred wheat. And so for this task, we are sequencing 12 highly diverse land races, plus we are resequencing Chinese spring. So these data will be released soon, and they will include not only complete genome sequences, but also will include transcriptomic data and gene annotations and an update to the RefSeq annotation. And then finally, all of the variation will be cataloged using a practical haplotype graph. So that's the plan for IWGSC going forward. In addition to the webinars, we also host each year a workshop at the Plant and Animal Genome Conference, and so that'll be taking place next month on Saturday, January 10th from 8 a.m. So if you're able to join PAG this year, we would be very happy to see you out in San Diego. Okay, we're just about to start, but I wanted to give a little bit of a course on the dashboard, webinar dashboard. The most important things are, well, first of all, this will be recorded and available on YouTube in case you want to see it again. If the organizers need to communicate with you, they'll use the chat. So, for example, that will include some PDFs and some other information that will be shared. If you have questions, please use the question Q&A button and ask your question. And people can also not only ask questions, but also can upvote questions that they want to prioritize. So there will be time at the end for questions. And without further ado, I'd like to introduce our speaker, Vijay Tiwary. He's an associate professor at the College of Agriculture and Natural Resources at the University of Maryland. And today's talk is entitled Ancient Grain Genomics to Simplify Gene Discovery and Deployment in Wheat. So thank you, everybody, and we'll see you at the end of the presentation. And thank you, Vijay.
Okay. Can you see my slides okay?
Yes.
Okay. Thanks, Josh, for this invitation and thank you for this opportunity to present my work here. I have been saying it long time that I feel that I'm kind of a child of IWGSC, and I've been very much involved in the all the meeting and discussions throughout my arrival in US in 2009, and I learned a lot through the interactions and work that this consortium was doing. Okay, so today I'm going to talk about my work on our group's work on einkorn genomics. As you see here a stand-up einkorn wheat in Maryland. They look pretty good. So I know this is a wheat consortium, and a lot of people are wheat users, are involved in the wheat genetics or genomics in a way. But still, it's a good reminder to the audience that wheat is one of the most important crop in feeding the world, and it sustained billions right from the inception of our bread wheat as a crop. It basically brought together us and help in developing the civilizations and established early trade routes, and then basically good or bad help and expanded the empires and created new worlds, brought revolution along with the evolution in the our increase in the brain capacity and our thinking processes. And you can see that it is still one of the most important crop on the globe, and one of the most important crop that when you look at the trade it brings to the globe. But along with all these cool things that we hear about wheat, we also know that uh, global wheat production faces some serious threat from a number of factors. We are looking at on the top panel how the heat is increasing across our planet, and then incidences of drought and flood, they're kind of keep increasing, and these instances are changing the global pattern, is basically making more inducive conducive environment for pests and diseases. So wheat has to basically keep the pace not only with the changing all these parameters and diseases, but also to basically reducing agricultural land. And I think we have been doing a pretty good job in keeping the pace in improving our crops, uh, and but still integrated and sustained solutions are required to basically making our crop plants more resource efficient. At the same time, we also need to take care of our soil health and maintaining whatever waste we are producing, and majority of the improvement that we are going to see is going to be coming from the better management practices and elite varieties that may have some solution to these problems. And genomics is a key component for solving some of the crops biotic or performance issues. And wheat the whole process of wheat genome sequencing has started way back, and I hear all the stories from Dr. Bikram Gill and Kelly that how the IWGSC was started in 2003 with the support from the Kansas wheat growers, and then the process have been in going on. And in 2014 the first draft genome of bread wheat came out, and in 2018 as a group we basically put together the gold standard wheat genome. And after that, we see an explosion in the genomics resource development in wheat. We have our pasta wheat genome available. We have wild emmer wheat genome that came a little bit earlier, around same time with the wheat genome. And then wheat's number of progenitor species and their genomes and pangenomes are available. Then wheat's pangenome came from the 10 plus project. So we can see that by end of 2024, we have major wheat progenitor genomes are available, and now with hundreds of wheat whole genome sequences and different genomes are available. So we can say that we are pretty good with where we should be with the genomics tools and resources in wheat. So genomics resources are sufficient. We basically hit the landmark there. Again, more is going to be merrier. But at this point, I think we also need to think about developing some cool tools and uh, arrival of gene editing tools and other modifiers and sequencing populations have basically helped in looking at genome wide and precise and to create precise genetic variation in wheat. So we can say that we are doing pretty good with the tool developments or tools that can manipulate genes and genome, but still we have a lingering limitation that with all these new tools and technologies we still feel that what are going to be our target if we look at in our last 10-15 years of our gene editing work, our gene manipulation work, we are limited by the target, which genes to edit, and that's where our in my opinion where our bigger limitation is. So the process of gene discovery needs to be a fast paced, and then we have other tools like speed breeding and double haploid to basically deploy those variation at faster pace in our cultivar development program. So when we look at the complexities of gene discovery in wheat, I think we have solved the problem of sequencing large genome. But wheat is a polyploid species and it has three genomes, A, B, and D, and they have very high sequence similarity at the gene level. So that basically create a functional redundancy. And this work is again, I'm oversimplifying this beautiful paper from Cristobal's group, where his data suggests that about 30-70% of the genes in wheat are basically have showing balanced triad. That means expression is very similar from A, B, and D genome specific genes from that. And about 18 to 20% genes are also controlled by two subgenomes, about 8 to 10% genes which are basically non-regulated by all these multiple genomes. So that means if you translate that information to basically dominant genes, maybe there are going to be 8 to 10% genes which are uh, showing single genome dominance and they can be targeted. So that's why about 90% of genes may not have a phenotype if you are just knocking down one of the mutant allele. That basically create a problem in gene discovery pipelines. And again, there's a very similar thing. You can see that A, B, and D genomes have their homologs and they're very similar. So that create a problem uh, when you want to discover the gene or when you want to fast track the gene discovery process in wheat, polyploid wheat. So some options are available in wheat gene pool because wheat is a allopolyploid. That means three different species came together and developed our wheat. So Eslavotoxia is the D genome donor of wheat. Has been very extensively used in the wheat improvement and breeding program. Then Eslavospeltoides, which is very close to the extent B genome donor of wheat, which is also used to some extent, not extensively, into wheat improvement program, and Tritcamuralto, which is a genome donor of wheat, but it lacks a lot of genetic diversity as other progenitor species in wheat. So Brande's group has Brande Wolf's group at Kaust has done a phenomenal job in using Eslavotoxia as a gene discovery model. On contrary, Titcum monococum, which is slightly different and evolves slightly differently from these three species, but present very unique set of genetic variation, which is not much exploited into the breeding program. And these it basically contains two species, the wild and domesticated one, and they have co-evolved in the same geoclimatic conditions. So that means they also bring together this beautiful and amazing co-evolutionary history between them that can shed the light of on our evolutionary pattern and help us understand the genetic adaptation of the crop. So today I'm going to talk about some ancient grain. They can be defined as the whole grains and seed that has largely remained unchanged for centuries. And these are basically have been cultivated and advanced because of their rich nutrient profile and unique flavors. And there are the list of all majority of the ancient grain that has been used, including einkorn, emmer, kamut, spelt, sorghum, and list goes on. Today, I'm going to focus on einkorn wheat. Why einkorn wheat? Because this is the first crop that humankind domesticated and first crop that we ate. And it is a cornerstone crop of our domestication, crop domestication. There's some evidence that is coming from this very unique site in Turkey, uh, Göbekli Tepe, and they found the abundance of einkorn grain, wild einkorn grains there, and there are number of articles that suggest that hunter gatherers they started feasting on some of the ancient grain, and einkorn wheat was one of the prominent sources there. And data suggests that they were still working with the wild einkorn wheat. And then there are some evidences suggesting that about 25 to 30 mile up north in northern Turkey, the wild einkorn was domesticated. So that shows some kind of trend that probably uh, at Göbekli Tepe, they saw the value of einkorn wheat and they started working together, and that's why maybe they domesticated einkorn wheat later in the northern Turkey. So that also shows very unique perspective of einkorn as our cornerstone crop. So Uh, again, some literature suggests that einkorn wheat was domesticated about 10,000 years back. Again, this is the wheat evolutionary chart that you can see that Ura to the A genome donor that got hybridized with the B genome donor. Uh, and then it gave our bread wheat about 10,000 years back, whereas this subspecies Azulpoides of monococcum got domesticated around same time 10 to 12,000 years back, and we got our domesticated einkorn wheat. But because it was not heavily used for the breeding, most of the uh, trait were our genes were still sustained in these species. The term monococcum comes from the coccum. Mono is one, and coccum is grain. That means each spikelet contains one grain, and it is close to the A genome donor of wheat. It has diploid genome. Genome is about 5 to 5.2 GB, and it has not been hybridized or modified. It has higher protein, vitamin, minerals than our cultivated wheat, and it provides unique texture and has weaker gluten content that can be easier to digest. So all these traits make einkorn a good crop to work with. And again, einkorn is being used in some portion in Europe and US as a crop too. So I think there's a potential to increase einkorn as a crop too, or advance as a crop model too. So because of its it is already an old crop, there's an interest in basically advancing einkorn wheat as a simple and elegant system for gene discovery in wheat. So the reason for that is that it has a simplified, simple diploid genome, and domesticated einkorn wheat has a spring wheat habit. And without even going for the speed breeding, we can easily take 4 generations of einkorn in a year. If you do speed breeding, you are talking about 5 to 6 generation. And in our group, I think we have everything that a geneticist can think for to basically establish a gene discovery pipeline. We have very large einkorn collection. We have reference genomes, mutant populations, and we have developed many mute map populations and sequence association panel. And uh, uh, Juan de Bernardi and George at UC Davis has also shown that transformation can be done in diploid A genome wheat, and you can look at the yield and economic component of this crop in the field condition, which is not feasible for Toshii, as of Toshii, and maybe for the speltoides because you cannot bring the grains back without shattering. So all these traits make Einkorn wheat an elegant system to perform gene discovery. So again, this was a grant that was funded to us from NIFA, and that allowed us to bring all these friends and collaborators to work together with an idea to generate some reference assemblies in Einkorn wheat, and then use our Einkorn association panel for gene discovery and deploy those integrations in wheat. So then I think the Jesse and Simon they started putting together Einkorn genome, and then we also joined the team, and we together put together one of the best known genome assemblies in Triticif. It was combining two unique accession Triticum monococcum subspecies monococcum, that is TA10622, that's a domesticated accession, and then TA299, that is a wild Triticum monococcum subspecies Azulepoides. And then because of very specific interest in two accession Triticum monococcum subspecies Azulepoides L95 and Triticum monococcum subspecies monococcum, that's a domesticated one, L96, we also assembled two genomes. So we had four different genomes, and we have basically established a very unique data set for Einkorn wheat. So you can go to this database and start looking at the relationship between Einkorn genes with bread wheat genes and Toshii and uh, other cereal genes, and there are, you can look at the synteny and all other relationship there. And this paper was also providing this the information about the database. So we try to put together all the resources in under one particular setup so we can fast track the discovery pipeline. So I will show you some examples how we are using monococcum as a model for gene discovery and translational research. So idea is that we have all these genomic resources and germplasm resources with us, and so we can basically do the gene discovery by getting to the all these mapping populations, mutant populations uh, in the Einkorn itself, and we can validate those candidate genes through simple tilling populations, transformation, or gene editing. And if we have to improve Einkorn wheat, we can identify those genes and use our elite monococcum line to improve that. And once we identify the genes here, we can go to Chronos or Cadenza tilling population because mutant information is already available. We just need to look into the bioinformatics of those homologous gene, and we can combine the mutant or see the effect of the mutation. And if it works in, in tetraploid wheat, then we hexaploid wheat, then we have gene editing to basically precisely create those useful mutations in bread wheat, or use PH1B based genetic transfer from diploid wheat to hexaploid wheat. So with this in mind, we started exploring our genomic and germplasm resources for basically gene discovery. We have used three independent approaches using GWAS, using our association panel. Then we have a sequence index allele population. And then we also developed multiple mute map population to validate those genes. And since we already have four genomes available, so we can basically track back and look at the genes and their uh, uh, gene profiles to basically make some decision what particular candidate genes to advance or not. So this was the information that was presented earlier that along with Simon and Jesse, we published first einkorn genomes of two uh, einkorn accessions. And you can see that with respect to Chinese spring reference genome, how improved the scaffolds are. Again, these are two different technologies and of two different ages. And Chinese Spring is still one of the gold standard genomes, and we follow that a lot. But you can see that how improvement in the technology has helped us in getting good quality genomes. And these are two other L95 and L96 genomes compared with the uh, wild and domesticated accession that we worked in our group. So we have about 1,500 Einkorn collection at University of Maryland, and these collection have been received from US, NSGC, from WGRC, NSGC, Canadian partners, and then some Israeli partners, as well as from IPK, Gattaspleen, Germany. So once we have those, we basically selected the lines that is representing the most genetic diversity. Excuse me. So we removed the duplicated accessions and identified a diverse set of about 320 accessions. Out of that, 218 accessions were already sequenced. So we added about 108 or 10 more accession to this particular list. So we have now 320 diverse accession with at least 10x of coverage. So, and this is the distribution of a set of those 320 lines, about 218 accessions. So you are seeing the distribution of wild einkorn wheat, which is uh, Triticum monococcum subspecies Azulopidis has three distinct races: alpha, beta, and gamma races. The beta races is basically highly less represented in this collection. That's why when we got the collection from our partners from Germany that enriched this particular race, so we have now very good representation of this race in our panel. What is interesting is that we followed Brandeis' paper from Nature Biotechnology, and we developed Kamer matrix, which has about 8,000 million Kamers. And when we looked at the LD distribution, we found that LD block for each chromosome are on an average about 1.150 kb. And in wheat or diploid wheat, you are looking at less than 5, 6 genes in that particular interval. So that really excited us. And we started doing some GWAS. And for phenotyping, basically, we went ahead and contacted global leaders in those diseases. For example, we contacted Dr. Beth Keller for powdery mildew, and Jim Colmer did for leaf rust, Jiamin Chen did stripe rust, Matt Rouse did for stem rust. And so we contacted all the experts across the globe to help us on these diseases. We are working on a manuscript, so I may not be able to show all the data, but just one example where I'm showing K-emergy was for stem rest resistance, and you can see that SR21 is already cloned for both the races. Here presented, we are basically confirming the clone genes, and then we are identifying some of the new loci that can be used in basically identifying new sources of resistance. So blue peak shows the resistance association, and red peaks are showing negative correlation for that particular trait. In addition to this GWAS panel, we also have a sequence indexed population. So it is a cross between a wild and a domesticated einkorn wheat. In both the cases, we have reference genome available. So all these reads can basically trace back to the reference genomes, and we can pinpoint the SNP or genetic variation at the genome level. So we basically use single seed descent, and we have about 1,600 real lines for this population, but we sequence about 812 real populations or real lines using skim sequencing. And because the parental lines have sequence information, so we can always go back and identify the parental SNPs and distribution the real population. So this figure is showing the distribution of parental alleles in the real population. And what we found is that about this population of 812 RILs have about 16,000 crossover breakpoint, and that basically bring our mapping resolution to up to 140 to 200 kV. And this basically mapping power has allowed us to basically perform some high scale genetic mapping of our selected trait. I will show you two examples. Uh, so Titcomb wild einkorn wheat has this red coleoptile color that you can see from here, and domesticated one has green. And the phenotypic difference is very clear. And because it is real population, so you expect distribution of 1 to 1 in the population. So we phenotype it, and then our distribution of the phenotype in the population was did not differ using chi-square test when we look at 1-for-1 distribution. So we found a major peak on chromosome 7A, and what you see here is the different recombinants here, and and this is the breakpoint. So what we found is that this mapping basically delineated the interval to about 670 kb interval. Again, we have not exhausted our mapping panel. We can still put more markers in there. So on an average, the mapping resolution is about 225 kb. We also use the GBS, this sequencing data to see the association score. And again, it is the same population, biparental population. So you are going to expect very similar pattern, but it also gave us very single strong peak on this chromosome 7A. And we had four candidate genes identified. One of them was most promising, and we worked on it and we validated the candidate gene. So we also use this population to look at some yield-related traits, and spikelet number per spike. Our fertile spikelet number per spike can increase the yield. So L96 has a lower spikelet number. L95, which is the wild einkorn, has a higher spikelet number. So we use this data and did the mapping after phenotyping the entire population. We found a very strong peak at chromosome 4A around 78 to 80 centimeter. When we look at it, we found that it is spanning about 300 to 490 kb interval, and it has only 3 candidate genes. So we are looking at the translation of this locus in tetraploid and hexaploid wheat, and we are also doing editing for these targeted genes. So I showed you the example how we are using biparental population for trait discovery. And then I will also talk to you about einkorn tilling population that we have. We have a tilling population of about 5,000 M2 individuals. And the mutation frequency is about 100 mutation per kb. And if we have the sequencing data available for this population, as we have for Kronos and Kerenja, then it can basically fix a missing link and basically fast track our gene discovery pipeline. So we took the randomly selected 12 M2 plant and we sequenced them using whole genome sequencing platform. And there are some stats, but in general, we are looking at only G2A SNPs. This blue uh, circle shows the distribution of gene density on the L96 genome, and this orange circle shows the distribution of SNPs or EMS-based SNPs on these chromosomes on those genes. So what we found is that the total number of genic mutation is about 1,000 to 1,100 mutation, which is about 2% of the total SNPs, and majority of the SNPs are intergenic, and in total we get about 58,000 SNPs per plant. If we translate that, that means sequencing of less than 2,000 individuals can provide us 70 mutations for any given gene. That can be a humongous resource for doing translational research in wheat. So now I will show you some examples how we have used this mutant resource to discover some genes. I'm going to show you some examples. We have already developed 16 mute map population in einkorn wheat that is related with some major traits are spikelet number, grain size, free thrashing mutant. So even domesticated einkorn wheat are not easy to thrash, and that is basically not a farmer easily trait. If we can make it a free thrashing line, then there will be more interest from the growers to advance this crop. So we are interested. We have one unique mutant that is basically very free thrashing monococcum. So we wanted to identify that gene. And then we are looking at some early flowering, some late flowering, and a number of dwarf mutants. And we also ask a question that how long it takes to get a gene. Again, this is the same pipeline. Once we identify the gene, how fast we can go and identify gene and deploy into our breeding germplasm. So again, if you look at the a set of population that we advanced from M2 to M4, we have identified about 110 mutants. Some of them are used for simply mute map population. Some of them have you used for delta plus based mute map plus pipeline. I will show you one example for tin3 locus that we also contributed to Simon and Jesse's paper that was published in Nature. So we had a mutant, we call it tin3 mutant, that gives just single tiller, unical mutant, and wild type monococcum gets you about 35 to 40 tillers. And these are some tiller primodia that show the single primodia is developing, whereas in wild type you can see these red dot that shows that multiple tiller primodias are showing up. So we cross these two mutants, this mutant with wild type, and develop a mute map population. And when we ran our mute map pipeline, we found a very strong peak on chromosome 3A toward the end of the chromosome 3L. Interestingly, there was a paper from Bikram's group, Bikram Gill's group from Kansas State, that has identified a locus that was contributing to tin3 phenotype. So it was good that we are hitting that range. And when we look at this unique locus, which was about 2.4 MB region, we found about 15 to 16 SNPs, but there was only one genic SNP. So we looked at that genic SNP, and it was a G to A SNP, which was toward the at the intron exon boundary. So we really got excited. We thought probably this mutation is creating missplicing variation. So and then we basically developed the marker out of that and ran it on a big population, and we found that it was always segregating with the phenotypes. That basically increased our confidence. And then we extracted the mRNA from the right stage, from the wild and mutant plant. And idea was that if it is a misspliced mutation right here, then probably we are seeing an intronic retention, and that is causing a truncation. So here you are looking wild type monococcum. You basically develop the marker that is flanking this region and this region. So because you are flanking it, the product size is going to be very small, about 100 to 180 to 100 base pair. But if you have intronic retention, then you will see uh, bigger bands. And that's what exactly happened. This is a copybook example of how intronic retention based truncation looks on the gel. So we confirm these mutants going back and identifying uh, more lines from the monococcum. But we also wanted to see how it will look into the bread wheat. So we basically did a blast, and we found that Jagger genome has this gene, and it is not truncated, and all A, B, and D copies are very similar. So we also have a Jagger tilling population. We did tilling for A copy, B copy, and D copy. We combined all those alleles together. It took us longer time than doing discovery in monococcum. So you can see that unless you have all three copies knocked out, even combining one or two copies doesn't help, because here you can see the mutant in A copy, mutant in B copy, and mutant in D copy. Even combining two does not help until you have a null mutation. So when we have a null mutation in Jagger, we get a single tiller. So that was very exciting to see that whatever gene we discovered in monococcum, that we will be able to do some translational research. Again, it would be interesting to see some more positive phenotype to make the story more stronger, uh, uh, to showcase with some agronomically important traits. But we were able to clone the gene from monococcum in 12 to 14 months, even including the mute map population development. But it took us about 16 to 18 or 24 months to basically combine all the mutant alleles because it is a winter wheat background. So then we are also did the similar approach for three independent mutant. This is tree threshing mutant. We are highly, highly interested in this mutant because that can increase uh, that can help the farmers who grow einkorn wheat. And then we also looked at two independent dwarfing gene mutants. Again, I'm not going to go into the process, but Einkorn wheat has 7 chromosomes. You can see that for free thrashing, we have very strong peak on the chromosome 2A. It's very close to the centromere. And then for dwarfing gene population 1, we have a very strong peak on chromosome 5AL. For dwarfing gene population 3, we have very strong peak on chromosome 2AL. So these are three independent population, and you can see that mute map is identifying very unique region. So, and when, for example, in this case, where you are looking at mute map pipeline, here because we are hitting the centromeric region, our mapping interval is big, about 20 MB region. But when we look into this region, because now we have four different genomes, we find that only 12 SNPs in the genic region are genes, and only two SNPs are deleterious. So we are basically working on some gene editing because we cannot go in bread wheat and identify and do editing to basically make free thrashing to non-free thrashing. So that's why we are working on to basically do editing in these two candidate genes, and we are working with Juan and Juan's group to make this transition happen. And it is going to be a pretty unique story for our growers. So this is basically, I showed you some of the work that we are doing with the gene discovery. But in cases, because I haven't shown you some of the results on our disease screening and GWAS and disease panel, when we found that majority of the genes that we have identified, they are shown up in these four reference genome 299, L95, L96, and 10622. In addition to these, Dr. Jorge Dubkowski has also shared some integration lines from DV92. It's a domesticated einkorn wheat, and we are basically identifying the integration in those. So we use this bridge cross scheme to basically integrate important einkorn region in our wheat cultivar. Again, I will not go in a lot of detail. Dalhai Ku from our co-PI from WGRC Kansas State has basically uh, painted the chromosome from where we made a cross with the tetraploid wheat with einkorn. So you can see that these 14 chromosomes are from einkorn, and then they were back crossed with bread wheat. And then he has used this PH1B mutant pipeline. So there will be some uh, recombination between a genome of monococcum and wheat chromosome. And these are some examples of the different integration lines. This is the einkorn wheat. This is wheat that was used to make cross. And you can see that there are some lines which are pretty much recovered. So we have about 800 to 900 lines that we have available to us from those four different genomes along with DV92. An idea is that we will do skim sequencing. We have already did a GBS on a couple of plates, and we still need to look at the data. But because we have been able to identify those integrations, those GWAS peaks, so we can look at those integration from specific genomes, and these germplasm can be out for breeders and geneticists for incorporating these into their cultivars. So this is where we are with the most of our gene discovery work in einkorn wheat, and we got amazing response from our friends and community to basically advance this work. So our team has got together with Dr. Gina Brown Gudera from USDA ARS Raleigh and Amanda, and we started a coalition to basically put together several INCON genomes. And then many friends and partners have joined us. So right now we have about 20. The goal of this whole consortium is put together reference genome of 26 ANCON genomes as telomere to telomere assembly, and then we will be developing pan-hap and haplotype map of all these 320 diverse ANCON accession, and we will develop an integrated pipeline by connecting these hap maps with 1500 ANCON wheat accession that we have using genomic imputation, and the databases will be available at Grain Genes, and seeds will be distributed through NSGC. Again, I know that these are very small numbers, so you may not read. So this is our collection of about 1500 INCON wheat, and based on the phenotypic data and genotypic information and diversity information, we have picked these diverse INCON accession for their selection, the pangenome. And I think I'm almost towards my time. So I would like to conclude by saying that we have seen, I know that I have not shared all the results with you, but we are convinced that INCON provides excellent resource for translational research in wheat from both perspective, improving wheat as well as improving INCON wheat. Work in progress on multiple genes that we have identified for their positional cloning and validation, and integrated germplasm resource and genomic tools will help us develop resilient wheat varieties in both the wheat. And a message for the community: if you want to join our consortium for INCON Pangenome, please join us and help us achieving the goals faster. And especially, I'm looking for some partnership to sequence our tilling population in monococcum. That will be a big community resource. This work would have not been possible without an army of students, postdoc, and friends and colleagues, and several of them are co-PIs on a number of grants. Um, all the students in my lab, as well as Dr. Nidhi Rawat's lab, who's a pathologist at an associate professor at University of Maryland. She does most of the disease work that we have showed here, and Brandy and Haneen and Michael from KAUST for their support and help, and from USD Albany, Dr. Lazo, Dr. Koo, and Tainer for grain gene related work, and good friends Jesse, Simon, Dr. Chuneja, Paula, Dalhoy, and Gray for helping me so much with my grant and helping all the time, and Martin and Manuel for helping with the Pangenome project, and Manuel for helping with all the annotations, and partners and collaborators all around, and WGRC for sharing some of the germplasm resources when we needed them. And at the end, I missed the logo for IWGSC here. I'm sorry about that. All the funders and collaborators all around. With that, I will stop here and will be very happy to take any questions.
Thank you so much, Vijay. What a tremendous talk. So much work you've done, and you've accomplished so much and really added so many resources to the wheat community. So we have a number of questions. Once again, people, members of the audience can upvote questions, and I will take them based on that prioritization. If we run out of time, you may be able to contact Vijay by email, and he has said that he's willing to answer questions and communicate that way. So to start, thank you for your nice presentation. My questions are, so there's two questions here. Can you clarify GWAS for biparental population and GWAS for natural population? Is GWAS possible for both?
Again, you can do it. I think if there's more information in the paper that we publish, you are basically looking at the market rate association. So we did that, but again, you are not going to get those. You are still doing the bipental mapping population. You are just using a different tool, but we wanted to see that whether both the approaches are bringing down to same loci. So if you have any major QTL or minor QTL, that might have showed up. But essentially, bipental population would be the preferred approach to look at it.
Okay, and the second question on seed size, is there a standard to classify seed size um, and the range, what the range is? Is it possible, is there a positive relationship between 1,000 seed weight and seed size, for example?
I'm pretty sure that there is, right? Because seed size, if you have a bigger seed size, it is going to be correlated with the grain weight too. So I haven't looked at it, but here the context for us was to look at the same wild type parent, and there was a certain mutation that might have increased the grain weight or grain size, right? For example, we know a couple of genes in wheat like TAGW2 in wheat and GW2 locus in rice. If you mutate that locus, then grain size, grain weight can be increased. So that was our rationale there. Again, there can be many things that can affect the grain size. For example, you have only a couple of spikelet in the spike, which is fertile, right? So most of the resources are going to be allocated to those spikelet. So that's why we need to be very careful that we look at the spike fullness, sterility, and then compare the grain weight and grain size.
Okay. Our next question, also two questions here. The first is, do you have any suggestions for distinguishing whether a strong Kamer peak reflects true gene presence absence versus or underlying sequence variation?
That's a very good question, and I think we have struggled because of this, because we made a decision that we got this gene, but when we started moving our KMR matrix to different genomes, we saw slightly different results. But I think in general, it should be okay because you are not looking at a single SNP, you are looking at a string of the sequences. So usually it works. But I think there could be some verification processes. Maybe you have all these KMers sequence information. You can also run some simple SNP matrix and see whether you are coming down to similar region or not. But we were a bit fortunate because we have multiple genomes and we have called those matrix on multiple genomes so we can correlate. So, but that's a very good question.
Excellent. And the second question is, is with regard to genome editing right now in Triticum monococcum. Is it currently working efficiently?
I will not say more, otherwise I have not taken the permission from Juan, so I will just keep my mouth shut. But I would say preliminary data suggests that it can work.
Okay. Okay, next question. Thanks for a great presentation. What are your thoughts and observations regarding root architecture, root length differences in diploid, tetraploid versus hexaploid wheat? Are there any findings, articles, resources that you recommend to have a detailed, that would show a detailed understanding of uh, the relationship or it, a detailed understanding of root architecture, including root length and uh, yeah,
I
between.
One of my master student, Mr. Ian Moot, he is looking at drought tolerance in einkorn panel, and but we have not compared that with the bread wheat. So since I don't have data, so I will not speak on that. But in general, we have seen lot of variation in the root biomass or root length in the monococcum panel. Uh, because there's a doses level between diploid and hexaploid wheat. So I expect, and it is basically established, that bread wheat may have more robust root system unless you are looking at toshii and growing into very constrained pot. But I would say I will read more on that. So yeah, I would not make any guess on that.
Okay. Okay. There's still quite a few questions. The next one, also thanking you for sharing your research. And the question is, uh, in SNP index analysis, what is the importance of negative SNP index peaks, and how should they be interpreted relative to positive peaks when identifying candidate QTL regions?
So you are talking about the GWAS Kamer matrix, right? So if what happens is, for example, when you are doing the GWAS analysis, a Kamer matrix, you are looking at a particular genome, right? Because your Kamer is going to be mapped to a particular genome, and then you are using that in the analysis. If that genome does not have a positive QTL, then you will see different color peak. That's what we have witnessed. So I think that means probably the genome or index genome that has been used as a source probably does not have the positive QTL for that. So, but we have encountered this problem a couple of times, but when we put it to the different genomes, we see the variation. So I think that's what it should incur to.
Okay. The next question regards interest, I think, in collaboration to work on soil-borne pathogens, but so maybe that individual can contact you
Yes.
directly on that. Um, Skipping down, we have time for maybe one more question. Um, how to bypass the evolutionary events exposed on hexaploid wheat genome while utilizing einkorn wheat for gene discovery?
Again, my argument would be that most of the complications from the evolutionary prospects can be neutralized if you just use a diploid system, right? So that's where the advantage is. The wild and domesticated species in monococcum, they have co-evolved. So whatever divergence we have seen in the radiation or adaptation is still there, and these have not been heavily selected for any breeding goal. So I think these two species, wild species, provide us gem of information to look into that and answer right here. In bread wheat, I think it gets a little bit more complicated because of very heavy selection, and so I would say that that's why the beauty of the diploid system to look into that, and we have this resource in hand.
Great. Okay, I think there's one more question. Again, it seems to be about maybe a question about collaboration. So maybe that individual can also contact you. We are just about out of time. I want to thank everybody who attended today for joining us and for the great questions. And again, thanks to our speaker, Vijay Tiwary, for really a terrific talk. And we will see you next time. So thanks again.
Thank you, everyone.
All right.